Let's talk about the megasporogenesis from sexual reproduction in flowering plants today. In our last video, we have discussed about microsporogenesis. In this video, we'll talk about megasporogenesis. Before I take you all to the how a megaspore is being formed, let us talk about what actually is a gynoecium. I have given the introduction in the introduction chapter, in the introduction part of the video as well. So, what is gynoecium? It's a free unit, of gynoecium. See, uh, you always know about stigma, style, and ovary, and all this together is called as pistil, and you can also call it as a carpel. So, this pistil or carpel is always one single unit. When you have a collection of carpel or a collection of pistil or a whorl of pistil, we'll all together collectively call it as gynoecium. So, what is a carpel and what is a pistil? It is a free unit of a gynoecium. Fine. So, what is carpel consisting of? You can see at the top you're having a stigma. There is a part that is connecting the stigma to the ovary, and this is called as style. So, we have stigma, style, and ovary. And inside the ovary, always remember that there is presence of ovules, right? You can see multiple ovules present over here. Now, what is a stigma? Stigma, you can see, is the topmost part. It's a part that is going to receive the pollen. It is a part that is going to catch the pollen. It will decide that whether the pollen will be allowed to enter inside or not. Next is the style. As I said you, it is a tube-like structure that is connecting the stigma and the ovary together, and the ovules together as well, which is inside the ovary. And what is an ovary? It is a basal part. It is gonna contain the ovule inside. After the fertilization, this is something that develops into a seed. So this is the whole structure of a pistil. Now, pistil can be one single, right? If there is one single carpel, like there is just stigma, style, and over, this is the one part, you'll call it as monocarpellary, where there is just one carpel present. There are certain flowers in which you can find more than one carpel. If there is presence of just two carpel, you'll call them as bicarpellary, and if there is presence of many carpels altogether, you will call it as multicarpellary. Now, when all the carpels are fused together, you can see that there are multiple carpels fused together, and this fusion is called as syncarpus. And you can see the structure in hibiscus, madar ke phool mein you can see the structure. As well, you can see the same in papaver. So all they are fused together, right? Fused in the form like this. See, they all are coming together, and you can see at the ovary point they are fused, right? 1 2 3 4 5 6, you can see. So this is called as syncarpus. On the other hand, apocarpus means, you can see this, right? They all call, the ovaries are basically free and the other parts are fused together. You can see this apocarpus ovary. See this, all the pistils are free here. This is apocarpus, it's an example of syncarpus. This is papaver, and this is hibiscus. These examples are important. Only the diagram sometimes is given in the question. You have to identify that this is the example of which plant. So monocarpellary, bicarpellary, and then there comes your multicarpellary, and a difference between syncarpus and apocarpus. Syn matlab when they are fused, apo matlab when they are free. They are not fused together, they can be separately. You can see over here. So this is apocarpus and this is syncarpus. Next is, what is a locule? So when you can see inside the ovary, the very important part of a pistil is we can having an ovary at the base, and this ovary inside is having ovule. But where is this ovule actually present? There is a kind of place in which it is present, and this particular place we call it as locule. It is a cavity inside the ovary, right? So what is this cavity basically made up of? This cavity is basically made up of parenchyma, and there is presence of a cushion present over there, and this is called as placenta. It is very clear that whenever we talk about placenta, you can always remember placenta in the terms of human beings as well, right? Which has a lot of things to provide to the developing ovule, and to the that ovule is further going to develop into a seed. There will be development of endosperm as well. So this ovule is made up of parenchyma, and this cushion is called as placenta. Now, there can be multiple ovules and there can be one ovule as well. So here you can see multiple ovules over there in both the cases, but their arrangement is different. So people might have learnt about placentation in class 11, the arrangement of ovule, about axile placentation, about parietal placentation, about basal placentation. So the arrangement of ovule in the ovary is called as placentation. Now the ovules can be one, can be many. So when there is just one ovule, remember the examples are wheat, paddy, mango, etc. Now, if there are many ovules, the example will be papaya, watermelon, and orchids as well. The examples are obviously important. Next, then what is a megasporangium? Definitely, inside the ovary, there was presence of ovule, right? There was presence of locule, and in this locule was a cavity. You can find multiple ovule over here. You know, this ovule is called as megasporangium, right? That one single ovule is called as megasporangium. Multiple layers are present, multiple coverings are present. Those coverings are called as integument. That is why we say that ovule is an integumented megasporangium. I have seen students getting confused into what is an ovule, what is a megasporangium, where is the ovule. I again take you from the starting, that this is the ovary. Inside the ovary there is presence of ovule, and this one single ovule acts as a megasporangium, because here there is presence of a megaspore. Fine. So what is an ovule? Ovule is only called as megasporangium. Now let us see the diagram of an ovule. You can see the structure in NCERT book as well. It's a very important diagram. Sometimes only the diagram has been given and the labelings here comes as fill in the blanks. Otherwise, a simple question of to just make a diagram of one single ovule also comes in CBSE examinations. So yeah, this is the picture you can see over here of one single megasporangium. Now just to see this stalk is, and this is a very simple diagram to be learned. This is called as funicle, right? This attachment part of the funicle with the whole body of the ovule is called as hilum, right? And if I take you to the whole body of the ovule, you can see this part is open, right? So anything can enter inside, specifically the water can enter inside from this place. This gap, this whole sort of stuff is called as micropyle, and always this ending is called as micropylar ending. On the other hand, you are having the opposite to micropylar end is always the chalazal end, and this portion is called as chalaza. Fine. These are the integuments. You can see outer integument, then there comes your inner integument. This cushion like place over here is called as nucellus. Nucellus, the role is exactly similar to tipitum when you people were learning about TS of anther. There was presence of tipitum which was giving nourishment to the developing microspores. Here there is presence of nucellus which is going to give nourishment to the developing megaspore over here. Fine. So this is the structure of a ovule. See the main body, it is covered with integument. This is the micropylar opening, this is the chalazal opening, right? This is nucellus that is made up of parenchyma, and this is going to give nourishment. It is having lot of abundant food reserve. Wherever there is parenchyma, know that parenchyma always reserves the food. It can easily undergo the process of photosynthesis for preparing the food over there. Embryo sac and female gametophyte is located, you can see here. This is the cushion like place, the cavity where there is presence of an embryo sac, right? This is an embryo sac. So where is the embryo sac present? So where from we have started this? We started this from the ovary. Inside the ovary there were presence of many ovules. This diagram is of one single ovule, and inside the ovule there is presence of embryo sac. So हम कहाँ से कहाँ आ गए? We started with the ovary. We further moved to inside, that is there was an ovule, and inside the ovule you can see that there is presence of embryo sac, right? So ovary, ovule, embryo sac. What is ovule? Ovule is a megasporangium. So what was there inside the megasporangium? This was a sac like structure. You were having an embryo sac. Okay? So before I take you to this specific picture of an embryo sac, this arrangement of ovule, this sometimes becomes important for us to know, that is ovule always arranged in this way? The answer is no. We can have multiple arrangements of ovule. In this arrangement you can see that this is the chalazal end, this is the micropylar end. They're always opposite to each other, and this kind of arrangement is called as an anatropous ovule. So what are the other types of arrangement of the ovules? You can see here, this is called as orthotropous, where this is a very erect arrangement. You can see the other one, you can see it is inverted, right? This inverted form is called as anatropous. This as well is inverted, but it is not completely inverted. It is half inverted, and I'll call it as hemianatropous. The other one you can see is, this is also a kind of inversion only, but here it is half inverted. You can see, as well as it is half inverted, the body is curved here, right? So this is called as compylotropous, right? Here you can see that the body, right, as well as the embryo sac is curved. In the everywhere else you were not watching the embryo sac getting completely curved, but here you can see the embryo sac as well is curved. So I'll call it as amphi means two, right? So both of them are curved together. So this is an amphitropous ovule. And then the funicle, here you can see, this was the funicle part, right? This was a stalk which was holding the ovule. This funicle as well is curved. So when the funicle is definitely coiled or curved, you will call it as sarcinotropous. So these are different types of arrangement of ovules that you have to study. Maybe not for the CBSE, but for the NEET examinations. So right. So orthotropous, that is, you can see very erect part, anatropous inverted, hemi half inverted, again half inverted as well as the curved body. Again both the embryo sac as well as the body of the ovule gets inverted, amphitropous, and when the funicle is coiled it is called as sarcinotropous. So these are the types of ovule. Fine. And this arrangement that you can see in your NCERT book, this is in which type of ovule? This is your anatropous ovule. Fine. So inside the ovule we were having the embryo sac. Now let us see that what happens here now. What is a megaspore? How is the megaspore going to develop further? So let us see here now that the process, what is a megasporogenesis? Now the formation of the megaspore, right, from the megaspore mother cell. This is exactly similar to microsporogenesis, where you have seen the formation of microspore from the microspore mother cell. That was called as microsporogenesis. Again, the formation of megaspore from the megaspore mother cell is called as megasporogenesis. So again, the megaspore from the mother cell is going to be formed. I said you earlier as well, the division over here is going to be meiosis, right? This happens due to meiotic only. So this meiotic division taking place over here will lead to the formation of 4 megaspore. Now the question is, where is this going to happen? Right, this is going to happen in the embryo sac, right? The female gametophyte, the embryo sac which you can see over here. You know, this is the result of what? This is the result of the megasporogenesis. So this embryo sac is the result of megasporogenesis. So how has this embryo sac formed from one single megaspore? This is what we are going to see. It means that inside the ovule there is presence of megaspores, there is presence of megaspore mother cell, of course. So inside the ovule you can see that there is presence of megaspore mother cell, which will undergo meiotic divisions, which will lead to the formation of an embryo sac. So let us talk about that one functional megaspore. Right, so this is, for example, the one functional megaspore. Again, look at over here. There is nucellus. This is a micropylar end. This is a chilazal end, and there was one functional megaspore. Right, the megaspore mother cell. So what will happen in this megaspore mother cell? This will undergo meiosis, and whenever it undergoes meiosis, the product will be tetrad. Right, there will be formation of 1, 2, 3, 4 megaspores. Initially there will be formation of dyad, and further they lead to the formation of a tetrad, and this is the result of meiosis. You will be getting 4 megaspores. Now similarly we have seen microspore tetrad in the microsporogenesis, right? And in that microspore tetrad, I said you very clearly that every single microspore has the potential to develop into a pollen grain, right? Every single microspore was having the potential to develop into pollen grain, but here not every single megaspore has the potential to develop into a megaspore, right? All of them are not having the potential to lead to the formation of an embryo sac. Only one will have, right? So what will happen of the three of them? The three of them gets degenerated, right? Three of them will get degenerated, they'll be dissolved, and only one will be left to form the embryo sac. In the microsporogenesis, all the 4 were having the potential, But here all the 4 are not having the potential. 3 of them will be degenerated. Only one will further form the embryo sac. Now how this one is going to further form the embryo sac? This will further form the embryo sac with the help of mitosis, right? With the help of mitosis, right? So when 3 gets degenerated and only one is having the potential, this kind of development is called as monosporic type of development. This is given in NCERT. It is very important. It is called as monosporic type of development. So the functional megaspores, it will further enlarge in size, and by the means of 3 successive mitoses. How? Mitosis 1, mitosis 2, mitosis 3. There was just one single megaspore, one mitosis, right? You can see divided into 2. You can see 2 nucleus over here. Further it will divide 1 2 3 4. Further it will divide 1 2 3 4 5 6 7 8. So this is gonna 8 nucleated embryo sac will be formed at the end. So 1 2 and 3, 3 successive mitoses have taken place, out of in which in this one functional megaspore. This leads to formation of 2 to 4, 4 to 8. Okay, so these are the 3 successive mitotic division, and this kind of development we call it as monosporic type of development. Now at the end you can see that how many nucleates is there? 1 2 3 4 5 6 7 8, right? Now again the cell division starts to form over here. When the cell division starts to form at the micropylar end and at the chalazial end, you can see that 4 nuclei they enter at the micropylar end, right? And other 4 will arrange at the chalazial end. It will be 4 to 4, micropylar end and the chalazial end. Now again the cell division starts to form over here. They have got migrated from the pole to the center as well. 2 will go, right? See, 3 over here, 3 over here, 2 at the center. This is how the female gametophyte, the embryo sac, is going to develop. Look over here, right? This was the functional megaspore. This leads to the formation of mitosis. Now this was the first mitosis, the result of first mitosis. You can see this is the result of the second mitosis. This is the result of the third mitosis. Now they'll start arranging themselves. Now the cell wall formation as well occurs. You know, all this phenomena is being given by S.G. Navaschin. Please note this down somewhere if you are sitting with a copy or something. S.G. Navaschin was the one who has given all this phenomena. Now before taking you forward, I'll take you to NCERT directly now. Right, look at the structure, right? This was the part, this was the megaspore mother cell, and we know it that in the mother cell what happened? Meiotic division happened. When meiosis took place in the megaspore mother cell, it led to the formation of megaspore tetrad, where I said you 3 of them got generated and only one was the functional megaspore. This one functional megaspore from here has undergone first mitosis, C2 nucleus, second mitosis, 4 nucleus, third mitosis, 8 nucleus, 3 successive mitoses. This led to the formation of the female gametophyte over here. Then what happened? The cell wall formation took place, and when the cell wall formation took place, the synergids were formed at the micropylar end, antipodals were formed at the chalazial end, and 2 nuclei moved towards the center, and this all together is a central cell with 2 nuclei, and we call it as polar nuclei. Now if I invert the diagram completely, you know, this diagram, if you invert it, the micropylar end will go at this end and the chalazial end will come to this end. So don't be confused into these 2 diagrams. This is just the inverted part of this. See, the chalazial at the top, the micropylar at the bottom, the same image. So where will the synergids go? At the bottom. See here, we're having the synergids over here, we're having the filiform apparatus. Fine. Then here you're having the egg, 1 2 3 nuclei, 2 at the center, and we call it as polar nuclei, 3 at the top at the chalazial end, and we'll call it as antipodals. So how many nucleus you can see here? 1 2 3 4 5 6 7 8. So this is a 8 nucleated structure. Now at the end the cell wall formation took place, right? So how many cells over here? 1 2 3 4 5 6, and this is a central cell. and inside the central cell you are having two nuclei and you call them as polar nuclei, right? So this central cell is 1. So now again if I start counting 1 2 3 4 5 6 and the last one, that is the seventh one, right? So we are having 7 cells over here and we are having in total 8 nuclei. So its the structure is called as a 7-celled 8 nucleated structure. See here, a typical angiosperm embryo sac is at the maturity will be having 8 nucleate and 7 cell. This is a very important stuff which you people need to remember. Fine. So this was enough for this video. In the next video I'll be taking you to double fertilization. What is double fertilization? Look at this anatropous ovule. It's a typical anatropous ovule present over here. Okay? This inside this anatropous ovule at the center, this was a nucellus. This is the embryo sac. What led to the formation of the embryo sac? Because here there is sporogenous tissue and there you are having a megaspore mother cell, and the megaspore mother cell underwent meiotic division and then 3 mitosis. 1 meiosis, 3 mitosis, 1 meiosis, 3 mitosis. Fine. And from that megaspore mother cell, what was formed? The embryo sac. This is the embryo sac that I am talking about inside the ovule where fertilization will take place. So this is the embryo sac. So what you can see at the end, this is a diagram of a matured embryo sac. Fine. So that's all for today. In the next video, we'll talk about double fertilization. Thank you.